welcome and thank you for being here today. I'm Navneet Singh, your host for this webinar. I know that today's topic might bring up a number of you may and want to let you know that we will address as many as we can in the time we have today. And I welcome and request Professor Dr. Deepak Balasubramanian to start this session. Welcome, sir. Over to you.
Yeah, thank you all so much for inviting me to give this talk. One of the important things that we get asked in the exam is can you elaborate the basic pathogenesis of head and neck squamous cell carcinoma? So, head and neck squamous cell carcinoma is slightly different from other solid organ malignancies, except for maybe the esophagus and the upper digestive tract, because the processes are quite dissimilar between the two. So this is a very basic presentation. I would try to finish it within the allotted time. And we'll just go through the different molecular mechanisms and the etiopathogenesis of head and neck cancer. My name is Deepak. I work at Amrita, and I'm part of the head, neck, and reconstructive team here. So the aims of this presentation is for all of us to understand the cell cycle, which is the basic thing that we need to know for understanding the different areas where the tumors tend to intervene and cause progression. We have to understand the hallmarks of cancer. We have to understand tobacco carcinogenesis, and of course, the viral carcinogenesis, which is now being described very often in literature. We have to understand the stepwise changes from dysplasia towards frank malignancy. We have to understand the genetic changes that happen in patients who have oral squamous cell or any other head neck squamous cell carcinoma. And we also have to understand the role of proto-oncogenes and suppressor genes when it comes to the malignant transformation of tissue. So the important thing for you to understand that tumorigenesis in the head neck area is a multi-step process. There are several steps that translate from the patient having dysplasia to CIS to frank malignancy. So it's a multi-step process with varying factors that influence the development of tumor. Because it's a multi-step process, there is an accumulation of multiple genetic alterations. It's just not one genetic alteration, but it's multiple genetic alterations which feed the tumorigenesis. And an important part of the initiation and progression of tumorigenesis is the genetic predisposition of the patient. Some patients do have impaired repair mechanisms. Some of them do have mutated proto-oncogenes to begin with. Some of them have defective DNA repair mechanisms to begin with. So these patients, or in quite a sizable number of patients, we have genetic predisposition. And then we come to the environmental factors. There are several environmental factors, including habits. You can start with dietary changes, with ultraviolet radiation, with smoking, tobacco, and altered sexual practices. So all of them fit into the environmental factors. Now, in the simplest form of tumorigenesis or etiopathogenesis, we have normal epithelium, which is influenced by exogenous factors such as smoking, alcohol, tobacco ingestion, smoked foods in case of nasopharynx. So you have exogenous, and then, aromatic dyes, polycarbons. So you have exogenous factors, and then you have endogenous factors, which is the predisposition. And there are three basic steps. There is an initiation stage, there is a promotion stage, and there is a progression stage. So the exogenous and endogenous factors work on the normal epithelium, and they initiate a change that results in epithelial hyperplasia. Once epithelial hyperplasia is attained, then there is a promotion of the epithelial hyperplasia to epithelial dysplasia. And once there is epithelial dysplasia, there is progression of that dysplasia into invasive cancers. And as you go along this pathway, you have genetic and epigenetic alterations, you have chromosomal aberrations, and you have different molecular interactions, mostly from the exogenous growth factors, unrestricted growth potential of tumor, and an inhibition to the restrictive or protective functions of the human genome. So there is initiation, there is promotion, and there's progression. And this is from normal epithelium to invasive cancer. This is the most simplest form of tumorigenesis that we can depict. So, how has molecular changes and how has molecular evaluation and understanding of tumor pathogenesis evolved from 1920 to the present day? So, it started with radiation included in the clinical practice. Then tobacco and alcohol were identified as risk structures. Then DNA structure was resolved. Then we started chemotherapy into clinical practice by the 1960s. And in the 1980s and 1990s, the HPV as a causative agent for cervical cancer and EGFR overexpression in lung cancer was found. Then, as we moved on, we found that HPV also causes head and neck squamous cell carcinoma, particularly oropharyngeal carcinoma. And then we also realized EGFR has been overexpressed in head and neck squamous cell carcinoma. Then we found out that there is a programmed death ligand was overexpressed in head and neck squamous cell carcinoma in 2000. And as we move into the 2020s, 30s, and 40s, we have identified the immune landscape of tumors. So far, it was the tumor alone, but now we have understood what the stromal microenvironment of the tumor and how it affects. We've also seen the impact of microbiome, that is the oral microbial involvement and the presence of oral microbiomes. And we also refined immunotherapy treatments, and we've understood hypoxia as an integral part of tumor response to treatment. And now we have tailor-made plans for patients who have HPV positive tumors, but mostly it's the immunotherapies that is going to move forward in the future. Now, you have to understand how the different exogenous factors affect the tumor progression and the way the tumor responds to treatment. So you have tobacco, alcohol, bacteria, HPV, and diet. And these are the different ways that these exogenous factors affect the mediators of tumorigenesis. So you have tobacco, which primarily increases EGFR, AKT, and mTOR. There's also upregulation of fibronectin and vimentin, and cytochrome P450 is dysregulated. And this results in tumor growth progression and induction of tumor growth and metastasis. Alcohol also affects vimentin, matrix metalloproteinase 2, MMP7, and MMP9. And this leads to chronic inflammation, which we know is one of the precursors for malignancy. And it also does induction of epithelial mesenchymal transitions or translocations. Bacteria have endotoxins, enzymes, and metabolic wastes. And the metabolic waste and their activation are pro-carcinogenic and cause drug resistance. HPV in diet causes genomic instability and mutation, and all of this affect the way that the tumor progresses and metastasizes based on the exogenous factors. Now, the basic premise in head neck squamous cell carcinoma or any malignancy that you see is that tumor suppressor genes are inactivated, So this can be done because this can occur because of either mutation, loss of heterozygosity, deletion, and epigenetic modifications. And tumor promoting genes are activated. And this occurs because of gene amplification. There is increased transcription of the genes and proteins, and this can be because of structural changes which occur in the or the promoting oncogenes. Now you have to understand the cell cycle very clearly. Basically, the cell cycle has four different stages, and all of you know it's the G1, S, G2, and M phase. The G1 phase is the growth phase for the cell. The S phase is where DNA synthesis occurs. The G2 phase is the growth phase and the precursor for mitosis. And M phase is mitosis, which is resulting in cell division. The picture on the right is more elaborate because you have a lot of subphases within that, like anaphase, metaphase, prometaphase, prophase, and the interface. So this cell cycle is very important to understand because different chemotherapeutic targets different parts of the cell cycle, and different proto-oncogenes or tumor promoting genes and proteins affect the different sites of the cell cycle. So this is a very simple view of the cancer. You assume that it's a mass or a lesion that contains cancer cells. And that is all that we think of as a tumor. And if the tumor breaches the tumor capsule or invades the lymphatics or vascular invasion, it becomes an invasive carcinoma when it leaves the primary site and becomes regionally metastatic or distantly metastatic. But the truth of the tumor is that it's not only the tumor cells, but the tumor precursor cells called the cancer stem cells. You have the body's defense mechanism, that is the immune inflammatory cells, which are in the tumor milieu. You have endothelial cells, which proliferate to form neovascularization. And the stroma is usually filled by cancer associated fibroblasts. So, this is a more complicated view of a cancer. It is just not simple cancer cells, but there are a lot of other cells associated within the cancer lesion. Now, this is a very important paper that I suggest all of you read. There has been multiple revisions of this paper. It's by Hanahan and Weinberg. It has got tens and thousands of citations in different journals and by different other authors. So it's one of the most important things that you have to read as an oncologist. So they found that there are different types of or different stages or different areas where the tumors affect normal tissue and become tumors. And they understood that tumor development proceeds in a way which is similar to Darwinian evolution, meaning that each successive cluster of tumor cells have successive genetic changes which confer growth advantage to these cells or leads to the conversion of normal human cells into cancer cells. And this was further set up in the way when the newer discoveries were made that mutations produce oncogenes with dominant gain of function, meaning that the oncogenes work more, and the tumor suppressor genes receive loss of function, meaning that the tumor suppressor genes work less. So these are the hallmarks of cancer as per Hanahan and Weinberg. So one is self-sufficiency in growth signals. So you don't need an extraneous growth signal to promote growth in these cancer cells. They have their own inbuilt mechanisms to give them tumor growth. There is insensitivity to anti-growth signals, meaning that whatever growth suppressor inflammatory signals come down, none of them work on the tumor cell. So there's an insensitivity to anti-growth signals. Then the tumor has the potential for tumor invasion and metastasis. This is done by circulating tumor cells which evade a capture by the immune cells. Then the tumor has limitless replicative potential because of the presence of the stem cells as well as changes in the telomerase of the cancer cell. And the most important thing is that they are able to sustain angiogenesis because without blood, there is no tumor growth. So they are able to sustain angiogenesis as long as the tumor remains in situ. And finally, they are able to evade apoptosis. So once you evade apoptosis, then you have limitless potential to replicate. So this is another important aspect of tumorigenesis. So let's look at each of these things in slight detail. So when you say that they have self-sufficiency in growth signals, they are autonomous. And this involves the manufacture of growth factors by the tumor cell itself. So you're not dependent on growth factors from other tissues. And this is well seen in glioblastomas, sarcomas, and even in head neck cancer. So platelet derived growth factor and transforming growth factor alpha are usually produced endogenously by the tumor. And then even if you don't have the growth factors, there is receptor overexpression. So even if you have less growth signals from the outside, because the receptors are overexpressed and the transcellular activation of the intracellular mechanisms, they are promoted because the receptors are overexpressed and the middleman also is overexpressed. And there's also an increase in the intracellular circuits that translate these signals into action. And one of them is the RAS-RAF MAPK cascade. So you either have increased growth factors by the tumor, or you have increased expression of the receptors and the middleman, that is the transcellular ligand. And then you have intrinsic overexpression of the intracellular circuits. So as I mentioned before, the stromal cells that occur along with the tumor are very important for providing these growth factors. So most of the growth factors driving the proliferation of the cancer cells originate from the stromal cell components of the tumor mass. And this is another picture. So you can see that there are different types of cells. And the most important are the tumor associated fibroblasts or the tumor derived fibroblasts. They are quite common in the cell milieu and they produce a lot of pro-tumoral substances like interleukins, NF, HGF, CTGF, CCL5, and prostaglandin-derived growth factors. And the others also work. For example, the lymphocytes also produce pro-tumoral mediator substances. Now, if you look at the different cell types, the different cell types have different functions, and this is a very exhaustive table. So you can see from the CD68 cells going down to the CD11, CD117, CD3, CD4. All these cells have an effect. This is the immune response to the tumor. Some of them promote tumor growth, some of them destroy tumor growth, but this is just a table to show you how much the function of the stromal cells are. So some of them promote angiogenesis and invasion. Some of them promote tumor progression. So CD68 and CD117 and CD11 promote all sorts of tumorigenesis. And CD3 and CD14 have cytotoxic activity. So these are some of the cells that modulate the tumor, the response of the immune cells to the tumor. And again, this is another picture showing the presence of the tumor milieu or the stromal background of the tumor. The second thing is insensitivity to anti-growth signals. So anti-growth signals can block proliferation by two different mechanisms. So cells may be moved out from the active proliferation stage into the gap stage, or they can relinquish their proliferative potential by inducing into a post-mitotic state. So this example is the retinoblastoma pathway in HPV oropharyngeal cancers. So these are the areas where the tumor attacks, and the anti-growth signals are nullified by the tumor and its metabolites. So these cells continuously proliferate because of the lack of anti-growth, the lack of effect of the anti-growth signals. So evading apoptosis is another important aspect of tumor. The most commonly involving is the loss of the pro-apoptotic regulator, which is the P53, which is a tumor suppression gene. So if you have a mutated, so there are two types of P53. One is the wild type and the other is the mutated type. Wild type is what exists and promotes apoptosis, and the mutated type is what evades apoptosis when it has been mutated. And it is seen in more than half of human cancers, and especially a lot in head and neck cancers. So if you have a tumor suppressor gene, it works by causing degradation of the tissues, and this can be brought about by DNA damage, hypoxia, heat shock, or ribosomal stress. So these are what arrests the cell cycle if you have a normal P53, but if you have an abnormal P53, there is dysregulated growth. So limitless replicative potential. Now, most of the cells will stop dividing, normal cells will stop dividing at a certain point and will undergo apoptosis, and they don't have limitless potential to replicate. And this is brought about by the progressive erosion of the telomeres. So each successive replication of a normal tumor cells cause a shortening of the telomeres by the telomerase enzyme, and the unprotected ends of these telomeres participate in chromosomal fusions if they are unprotected. So there is maintenance of telomerase, which results in tumorigenesis, and it is seen in virtually all types of tumor cells. So there is maintenance of telomerase in these cancer cells, and this results in tumorigenesis and is quite commonly seen in head and neck cancers. So I told you sustained angiogenesis is one of the important aspects. Tumors are, they can do an angiogenic switch. So by changing, so there's always a balance between the angiogenesis inducers and the inhibitors. But tumors are able to change the balance between these two. And this is done by overexpression of VEGF or fibroblast growth factors compared to normal tissue counterparts. And there's also the inhibition of endogenous inhibitors like thrombospondin and beta interferon. These are downregulated, and these are some of the targets that people are trying to use as a therapeutic target for inhibiting tumorigenesis. The next is tissue invasion and metastasis. Usually cells are tethered together to their surroundings, and this is what forms normal tissue organs. But these cell-to-cell adhesion molecules and the adherence of these cells is affected in tumor tissue. And these are mostly, the affected proteins are mostly cell adhesion molecules and integrins. Now, when you lose the cell adhesion, the tumor tends to dissipate. There results an invasive tumor microenvironment. And from this microenvironment, the tumors can enter into the blood vessel and lymphatics, and it results in a metastatic tumor microenvironment. So what basically happens is there is cancer cells. The cancer cells become invasive because of loss of cell adhesions. And then there is recruitment of the stromal cells, which are needed to seed the metastasis. And then there is macrometastasis. And this process is repeated till the second wave of metastasis occurs. So there are various examples. So the cadherin coupling between adjacent cells by E-cadherin results in the transmission of anti-growth signals, but that is lost. And there are several enzymes like proteases that also factor into these metastases. So if you look at the genetic changes that we have in head and neck cancers, P16 inactivation is seen about 70%. P53 is in about 50%. HPV integration in 25, we'll come to HPV a bit later. P13 AKT mTOR pathway is in more than 40%. VEGF and other pathways, along with cadherin and matrix metalloproteins, are variable, and these have different functions on the tumor. Now, I mentioned to you that P53 mutations are the most, or one of the most common mutations in head and neck squamous cell carcinoma. This is a very important article by Poeta, where they looked at P53 mutations and survival in head and neck squamous cell carcinoma. So the wild type P53, which is what normally occurs, leads to growth arrest, apoptosis, prevention of angiogenesis, and helps in DNA repair. Whereas the mutant P53 has loss of function or gain of function. So if there is loss of function, none of the above mentioned things occur. When there is gain of function, there is proliferation, survival of the tumor, metastasis, and drug resistance. And people who have P53 mutations have less survival compared to people who have the wild type P53. And this is one of the reasons why HPV has better outcomes, because it doesn't cause mutation of P53, and the wild type is still available to do all the things to prevent the tumor. Now, looking at tobacco-related carcinogenesis in head and neck, which is the most common exogenous factor. So you know that there are carcinogens, and in tobacco smoke or any other form of tobacco, there can be two outcomes of the metabolism of these carcinogens. There can be metabolic activation, or there can be metabolic detoxification. Detoxification results in excretion of the metabolite, and activation causes DNA adducts. And again, there are two outcomes of these DNA adducts. Either they can be repaired to form normal DNA, but when these DNA adducts persist, there can be mutations in RAS, TP53, and other genes. And then there is loss of normal growth function, and there is carcinogenesis. The cigarette smoking can go without the DNA adducts by promoting tumor and gene promoter methylation. There is tumor suppressor inactivation and enhanced carcinogenicity. Or the carcinogen can bind with a receptor, activate the proto-oncogenes, and result in loss of apoptosis, increase in angiogenesis, and transformation. So cigarette smoking has three main pathways: either receptor binding, or tumor promotion, or DNA adducts. Now, these are some of the chemicals within the cigarette smoke that has been identified as causes for, or as a carcinogen. So you have polycyclic aromatic hydrocarbons, you have nitrosamines, aromatic amines, aldehydes, various different types of hydrocarbons, and other organic and inorganic compounds. Now, if you look at the biological or the molecular level of how tobacco affects biology, let's say that we are talking about the genetic alterations. I mentioned there are three pathways. In this, I will discuss two in detail. So the initial genetic insult causes deletion of 3P and 9P. And as we move from normal mucosa to dysplasia, we have further methylations, further amplifications, and loss of heterozygosity at different genes. And then once we move from dysplasia to cancer, we have P53 mutations and 13q and 18q deletions. Now, the other pathway is when there is mucosal damage and there is inflammation. So there is COX2 activation after mucosal damage. There is EGFR activation. There is cyclin D1 activation, increased proliferation. And once this happens, once there is more proliferation, there's always a higher chance and higher frequency of mutation. So this is the genetic level of how tobacco affects mucosa and the genes. So when you look at HPV 16 and 18, which are now implicated in oropharyngeal carcinomas and some sinonasal malignancies, there are two basic oncogenes, E6 and E7, and they act in two different ways. One acts via P53, and E7 acts via the retinoblastoma tumor suppressor product. So what happens is both of these, one, the P53 is degraded, and E7 releases the transcription factor E2F and facilitates cell cycle progression. So here, as I mentioned, there is no mutation of P53, only degradation. So whatever wild type P53 is present will act during DNA repair following treatment, and that's why these patients have better outcomes compared to tobacco-mediated SCC. Now, what are the other genetic alterations? So the most common genetic alteration in squamous cell carcinoma is loss of 9p21. And this is seen even before the tumor. It occurs in 70 to 80% of dysplastic lesions. Then there is a CDKNA2A locus, which encodes for tumor suppression P16 and P14, which are frequently inactivated by hypermethylation. So along with 9p, there's also loss of 3p, and this is again caused by hypermethylation, and these promote tumorigenesis. So another thing that we have to look at dysplasia, I told you that 9p21 is seen in dysplasia in about 70% of patients, but there's also amplification of 11q13 and overexpression of cyclin D1 in about 40% of cases of squamous dysplasia. And one of the early markers of loss of chromosomal material in dysplastic lesions, which are early markers of carcinogenesis, are 9p, 3p, and 17p. And if you look at 13q and 8p, they are more frequently seen in carcinomas than in dysplasia and are associated with later stages in carcinogenesis. So you've got to remember the deletions and overexpressions in dysplasia, and you've got to remember the losses of material that are seen in dysplasia, and you've got to remember the losses of material that are seen in carcinomas. So how do you see when something is going to progress from dysplasia? So the progression is seen when there's loss of heterozygosity of 17p and mutation of P53. And alterations of P53, whether it's a mutation or deletion, leads to increased genomic instability and may accelerate the genetic alterations in oral carcinogenesis. Now, we don't do these things routinely in clinical practice, but these are some of the markers that tell you whether a pre-malignant dysplasia, a pre-malignant lesion or dysplasia is going to transform into a malignant lesion. Now, the other important thing is field cancerization. So you know that field cancerization is multiple oral cancers. They can be from separate independent cell clones, or more recently, they can be from the same cell clone which has migrated. The hypothesis has been supported by mutation analysis through inactivation studies, microsatellite analysis, and P53 mutational studies. And these tumors are usually clonally related and derived from expansion of a single clone. Now, if there's a paper by Brackquist where the second primary, they have classified the tumors into second primary tumor and second field tumor. I suggest you read that paper. It's in Head and Neck. It is probably the current accepted terminologies for second primaries, synchronous and metachronous tumors. So usually what happens in field cancerization, there is a progression model. So there is a stem cell in the basal layer of epithelium, and it results in a clonal unit consisting of the stem cells with daughter cells, and these share the DNA alteration along with the remaining cells of that unit. Now, this patch progresses into an expanding field as a result of additional genetic change. So this is a progression model. And there's another model, which is a migratory model, which has the same basic concept, but it is separated in a greater distance from the original tumor cell. Now, what are proto-oncogenes and oncogenes? So oncogenes can be classified into functional groups. So there are growth factors or growth factor receptors. There are intracellular signal transducers. There are transcription factors. There are cell cycle regulators, and there are some mediators which inhibit the apoptosis of the cell. Now, the different cell types, the cell receptor types are the epidermal growth factor, that is the most common. And the most common down pathway is the ERB group of receptor proteins. They participate in multiple signaling pathways, and this occurs because of phosphorylation of the inner aspect of the receptors. And they can work in the RAS pathway, the SRC pathway, or the P13K pathway. And all of this down cascade results in cell cycle progression, survival, and proliferation of these tumor cells. So this is another more elaborate diagram, and this is in context with platinum therapy in head and neck cancers and cetuximab. Cetuximab works against EGFR. It blocks the EGFR receptor, and hence the down pathway or the cascading pathway is lost. And this will help to have tumor growth, suppression, and improvements in survival. But if EGFR is constitutionally activated because of the head and neck squamous cell carcinoma, there is tumor growth, survival, metastasis, angiogenesis, and invasion. And we have different types of receptors. We have the GPCR, we have the MMP, we have the EGFR, we have the TGF, and we have the VGF. So all of this causes changes in the intracellular pathways and promote tumorigenesis. So one of the targets for chemotherapy which was tried or which is still being used is EGFR blockade using cetuximab. So one of the pathway inhibitors works at the level of the extracellular binding area of the receptor. But there's also another target, that is a tyrosine kinase target. And we have different TKI inhibitors also, which are used for chemotherapy in patients who have tumors that are progressing in spite of standard treatment, or recurrent or metastatic tumors. Now, what is the role of the oral microbiome? Now, this is gaining importance. And as I showed you in the progression chart over the years, how things have changed. And now we're looking at hypoxia products. We are looking at oral microbiome. We are looking at immunotherapy. So the oral microbiome is basically the bacteria that you have in the mouth. And these have implications in either tumor progression, or they have implications in response to tumor treatment. So there are various different organisms that have been identified and thought to be promoting tumorigenesis, and there are some organisms which prevent tumor progression and enhance the response to tumor cells. This is an evolving concept. It has still not been studied in detail to make consensus, but you have to understand that the oral microbiome, and this also has implications in the way the patient responds to radiation. So these are some of the bacteria. So you have Capnophagia, you have F. nucleatum, then you have Staph serinomonas. Again, we have nucleatum again. And these are all the effects of these bacteria. So most of them are unfavorable to survival and to disease-specific survival, and some of them are favorable. Again, these are all preliminary studies that have been done. More work is going to be published, and then we would understand which oral microbiome is protective and which is not protective. Now, if you look at the oncogenic viruses, we look at proteins in microbiome, but we have to look at, they are not oncogenic. I mean, their DNA is not incorporated into the tumor mass or the tumor gene or genome. But these are some of the oncogenic viruses where there might be incorporation into the host, the tumor genome. Now, the commonest things that we know are HPV positive and negative and EBV. But in recent times, hepatitis C and hepatitis B have also been implicated. And hepatitis C and B usually cause squamous cell carcinoma or adenocarcinoma. And hepatitis C especially has a 6.7-fold increase in male patients for squamous cell carcinoma of the oral cavity, oropharynx, and larynx. And E6 and E7, and you know that from the HPV positive and negative tumors, and EBV is for the nasopharyngeal carcinoma. Now, there's another aspect called microRNAs, and these have been described very commonly these days. So, microRNAs can be detected in various bodily fluids, and there are two types. One is the tumor suppressor microRNA, and there's a tumor promoter microRNA. So, the tumor suppressor is downregulated, which causes increase in the oncogenes, whereas in the tumor promotion oncogenes is upregulated, and that causes inactivation of the tumor suppressor genes, and both of these lead to carcinogenesis. Now, these are the different types of microRNAs that have been found in head and neck squamous cell carcinoma. Now, as I mentioned, they can be detected in saliva, tumor tissues, serum, plasma, or peripheral blood. And each of these rings demonstrate where they have been isolated from. So this is a new area where people have started looking at the promotion or suppression of the oncogene by the microRNA. Now, another interesting aspect is DNA damage caused either by exogenous factors or endogenous factors, and the way the body repairs this DNA damage. Now, this DNA damage leads to DNA repair pathways, and this can lead to dysregulated proteins. Now, these are potential targets for drugs because once you give these drugs, these cells die out. They don't progress into abnormal pathways to form tumor cells. So these are some of the targeted therapies. So even tumor DNA damage and DNA damage to cell death, those pathways also can be altered in squamous cell carcinoma, and these can be blocked by using targeted therapies. So these are some of the dysregulated genes in the DNA repair mechanisms. So you see that they have different clinical effects. So all of these can be blocked by using targeted therapies or can be upregulated to enhance the response to chemotherapy. But most of them are downregulated, and you can actually block these by using targeted therapy. So in conclusion, head neck squamous cell carcinoma is a multi-step process. There's accumulation of multiple genetic alterations. There is a genetic predisposition for these patients, and environmental factors play a huge role in the initiation and progression of oral squamous cell and other head neck squamous cell carcinomas. Thank you for your time, and I hope all of you had understood a bit of basics of pathogenesis of head and neck squamous cell carcinoma.
Thank you very much, Dr. Deepak. Please, you may type your questions, comments in the chat box.
Let me open the chat box.
You may stop sharing the screen now and open the chat box on the Q&A.
There is no questions in the chat box. This is a very, yeah. Sir, can tumor infiltrating lymphocytes use as a prognostic factor? And what is the scope for targeted therapy to them? So, I don't know if you've read literature where they have looked at circulating blood cells like lymphocytes, neutrophils, monocytes, and they have found ratios which are having prognostic implications for survival. Now, there is no way to target a lymphocyte, but lymphocytes have been used, serum lymphocyte markers and ratios have been used for prognostication. There are many papers, we have also published papers on that. But within the tumor milieu, you can't target them. Now, the second thing is you can have them. There are different scores like Brandwein and all the other scores for pathology where they have spoken about lymphocytes infiltrating into the tumor. And if they do infiltrate, it's considered to be a good prognosis rather than a worse prognosis. So, pathologically, you can analyze, but there's no way to target them. But you can use serum ratios, NLR, MLR, different types of ratios to prognosticate the patient. The next question is, sir, as this decade is exploring immunotherapy, we have seen an association of tobacco and increase in PDL1 expression. PDL1 expression is not specific to tobacco or anything. It is specific to the inherent cell pathway. So probably because we see a lot of patients with smoking that come with PDL1 expression, but I wouldn't use PDL1 as a prognostic thing to say that if a patient has PDL1, when you're starting conventional therapy, that it's got anything to do with outcomes. So, yeah, you will see a lot more because we are testing a lot more PDL1 patients in unresectable and metastatic. But if you look at the whole cohort, I don't think there'll be any sort of association between the two. I hope I answered the question. How many of you have experience with PDL1 and head neck? I mean, is there any candidate here who has seen a lot of PDL1 treatment? Sorry? Hello?
Yes, sir.
No, how many of you have had experience with PDL1 treatment based on the PDL1 expression? So do you have a cutoff for the expression or do you any PDL1 or any PDL1 expression is? So there's a very interesting question. CA tongue in non-tobacco chewer and young people, any difference in mechanism? No, it's the constitutional absence of all the tumor suppressor genes. That is the reason that these patients have tumors. And we have published a lot in this area. There's a lot of papers. There's no consensus into whether these patients do better or worse. One argument is that these patients do worse because they live to see second tumors and other primaries, and hence their outcome successively becomes worse. But the only difference in mechanism is constitutional P53 mutation and loss of cell inhibitor mechanisms. But the outcomes is very varied. I've seen some who do very well. I've seen some who don't do very well. We didn't find any difference in our data, which we published, but other centers have shown worse outcomes for younger patients. So Only difference in mechanism is P53.
Thank you, Dr. Deepak. That was an excellent talk, and you almost answered all the questions that have been put there. Tough topic, but very nicely summarized. Thank you very much, and a fantastic, introduction talk to the beginning of our series. Thank you very much for taking your time in such a busy schedule. Thank you, and heartfelt thanks on behalf of DNB board, myself, and the students. Thank you very much, and over to Hnscc.
Thank you very much. Thank you, sir. It was very interesting to present this, and I hope all of the registrars and the fellows all were able to understand a bit of this topic. And if any of you want specifically anything, you can mail me. My mail ID is deepakbala@live. I'll type it out in the chat box. If you need any papers or anything at all, which you're not able to find, it's a learning experience for me also. That's how I look at it. Even for this, I learned a lot when I was preparing. So if any of you have any papers that you need, or if you need any information with academics, I'm sure clinically you guys are all great. Anything with academics or any papers or anything, we have a huge collection actually in our for all our fellows, for all our MCH residents. There's a 60 GB package worth of textbooks and papers that we have. If the organizers, Som sir, and everybody find a way, it's not legal in the strict terms, but if any of you want this material, I'm more than happy to send a pen drive across, and all of you can just get a copy of those textbooks. It's only head, neck, and plastic, but it's very useful to have.
Thank you. That would be very useful. Thank you very much.
Yeah, thank you very much, Professor Dr. Deepak Balasubramanian, for the presentation, and thank you very much, Professor Dr. for joining. Thank you, faculty members,